The human brain is a complicated organ made up of networks of neurons used to transmit signals back and forth. In simple terms, it is the organ responsible for telling the body what to do and when to do it. In an ongoing attempt to further understand just how the brain operates, scientists have been trying to chart the connectivity and functions of neurons in the brain, a task that is as fascinating as it is challenging. Now, with a new method developed at MIT, researchers have a new way to image brain tissue at not just one, but at multiple scales, allowing them to peer at molecules within cells or take a wider view of the long-range connections between neurons. Their new technique, known as magnified analysis of proteome, or MAP, uses a chemical process to expand tissue samples while preserving all of the proteins within the cells down to the nanoscopic details. Their technique relies on flooding the brain tissues with acrylamide monomers, which are attached to the proteins using formaldehyde. The researchers then link the monomers together into a dense, naturally expandable gel. Once proteins are denatured and separated, the gel expands the tissue sample to 4 or 5 times its original size. Once the tissue is expanded, the researchers use an off-the-shelf antibody to fluorescently tag specific proteins they'd like to image. Then, using a microscope, the researchers are able to obtain images with a resolution as high as 60 nanometers, much better than the usual 200 to 250 nanometer limit of light microscopes, which are constrained by the wavelength of visible light. The researchers also showed that the technique is applicable to other organs, such as the heart, lungs, liver, and kidneys. Current efforts to map the connections of the brain rely on electron microscopy or low-resolution light microscopy. But this team of MIT researchers have demonstrated that the high-resolution map imaging technique can trace neural connections easily and more accurately.